Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Nonhlanhla Mtshali. The lesson is proudly brought to you by the Gauteng Department of Education. Please remember, throughout this lesson, you can be in contact with us through questions. If you want to ask questions, please make use of the following number, 064 748 8913. This is our WhatsApp number. I will repeat the number again. 064 748 8913. Okay, I'm going to start with the topic for today. It's Life Sciences revision for paper 2, and we're going to look at the content that you need to prepare for your paper on Monday. So looking at the content for paper 2 and the weightings, we have DNA code of life, which is 27 marks. We have meiosis, which is 21 marks. We have genetics and inheritance, which is 48. We have evolution, which is 54 marks. Now, looking at the structure of the question paper. Remember, the paper that you're going to write on Monday is the same as the paper that you wrote on Friday, can't remember the date, but the first paper of Life Sciences that you wrote now in September. It is made up of 2 sections. You've got section A and section B. Section A is out of 50 marks. Section B is out of 100 marks. Our paper in total, it is out of 150 marks. And the important documents that you're going to need in preparation for this exam. Remember that you need to go through your past question papers, and you also need to look at the exemplar papers for 2021. There's 4 sets of those papers, and also papers from other provinces that have already been written. These you will get from your teachers. Also need to look at your exam guideline and the different textbooks, our DBE textbook. Also your Mind the Gap and SIP material, teacher notes and activities. If you do not have access to the past question papers, you can get all the question papers that are related to life sciences from this website, www.education.gov.za. We're going to look at the marking principles we use at the end of the year. So for example, let's say the question is asking you for the 3 reasons why this has taken place. If you as a learner decide to write 5 reasons instead of 3, we are going to mark the first 3 reasons, regardless of whether those reasons are incorrect. You need to give 3. So what is important here is read the question carefully. Make sure that you understand the question before you respond. Also, if the question has asked you for the comparisons, it is important that you need to write what is expected, the comparisons. If you've written your descriptions, if you've given descriptions, those will only be accepted if they are clear. Thirdly, if the question has asked you to tabulate, that is exactly what we are asking you to do. Draw a table, put the heading on the table, and then tabulate. If the question is tabulate between RNA and DNA, you draw your table, you write RNA, DNA. For the fact that you have drawn the table, that's a 1 mark already. So do draw your table, put your headings, even if your differences you are not sure of, but the fact that the table is there, you've already scored yourself a mark. If you are asked to describe and you've given a flow diagram, if the question says describe, you decide to answer in a form of a flow diagram, also you're not going to get marks. Marks will be lost. If it's multiple choice questions where you're supposed to give a letter, or it's a diagram where they say provide letters of the structures that are performing these functions, you give the letter, not the name. You lose marks if you give the name instead of the letter, or vice versa. And then let's start with our topics for today. DNA code of life. This is it's the section that you will find in paper 2. It is 27 marks, which is 18% of the question paper. The possible questions that you will get here, obviously, it's your terminology that you will find mainly in section A. You will get the questions where you're supposed to interpret diagrams. You will be asked to tabulate the differences. You might be asked to describe the processes that are taking place. You might also be asked to do some form of calculations. With meiosis, it's the same thing. There's terminology, there's interpretation of diagrams, there's tabulation, and there's also differences between meiosis and mitosis. There can also be differences between meiosis 1 and meiosis 2. Now let's look at the content itself. So for DNA and the RNA, what I said is important as it is identified on your exam guideline. It's the structure, the location, and the functions of the DNA and the RNA. And then we have this process, which is very important, DNA replication. Again, this is the process where the DNA makes the copy of itself that can be shared between the resulting daughter cells during cell division. Also have DNA profiling. This is also important. You've seen a lot of questions already coming from this section of the topic. And then we also have protein synthesis. Protein synthesis, especially the first one, transcription, this is a commonly asked question. So these are the things that we're going to look at. Moving on, the structure of the DNA and the RNA. Here they are put side by side for you so that you can see the differences between the two. We have the DNA and we have the RNA. The DNA, when you look into it, it is made up of 2 strands. In its natural form, it is a double helix structure. And then we have the 2 strands that are held together by the hydrogen bonds, as you can see there. When you look at the RNA on the other side, it is just a single-stranded structure and there are no hydrogen bonds there. The DNA is made up of the deoxyribose sugar. It is a double helix structure made up of double strands. It is usually longer than the RNA, and it is made up of the building blocks that are called nucleotides. Moving on to the RNA. RNA is made up of a ribose sugar. It is a single strand. It is usually shorter than the DNA, and it is also made up of the building blocks that are called nucleotides. By the way, the building blocks, we call them the monomers. So when we talk about these large molecules that are made up of smaller units, the smaller units are called the monomers. Then the monomers differ. For the DNA and RNA, the monomers are called the nucleotides. For example, when you look at the sugars, your monosaccharides, disaccharides, and polysaccharides, we call the monomers the saccharides. So those are the things that we look at. Moving on. Now, the building blocks. We said the building blocks are called the nucleotides. So for each nucleotide, whether it's in the RNA or it is in the DNA, it is made up of a phosphate, it is made up of a pentose sugar, and it is made up of a nitrogenous base. Again, these are the questions that you will get in section A. These are the questions where you'll find when you are given the structure of the RNA, you're supposed to identify the parts that make up the structure. Then this is where you need to get the information. Then the nitrogenous bases, there is 4 of them in the DNA. The DNA has the guanine, cytosine, adenine, and it is made up of the thymine. Please take note, in the RNA, the thymine is replaced by the uracil. So we have your uracil and your thymine. And at the bottom there, on the pentose sugar, the DNA is made up of, the DNA consists of a deoxyribose sugar, and the RNA consists of or has a ribose sugar. Hence the names DNA and RNA. DNA for deoxyribose, RNA for ribose. So this is the deoxyribonucleic acid, and this is the ribonucleic acid. So these are the nucleotides, the building blocks of the DNA and the RNA. Now, the important part, DNA replication. This is a commonly asked question. You'll be asked to describe the process, or the process will be there in front of you, and then you'll have to identify what process is this and where does it take place. So when we talk of a DNA replication, it usually takes place before cell division could occur, so that the DNA doubles itself. It makes the exact copy of itself. So what do you see here? This diagram, this is what we find on our Mind the Gap. So when you study, it is important that you make use of diagrams such as this, where there's only keywords that are given to you. You can see that there's unwind, unzips, there's template, and then there's complementary strand, and then there's two DNA molecules. So here, important is the description. And also, please pay attention. Your DNA replication and transcription in protein synthesis, those are usually mixed up because they are more or less the same. So it is important that you pay attention and you actually read the question so that you know whether is it asking me to describe DNA replication, or am I being asked to describe transcription. So when the process of DNA replication starts, we say the double helix unwinds. As you can see there, it unwinds. And when it does that, what happens next is that the bonds that are holding the two strands together here, they will break, and then the two strands separate. And after that, each original strand, now we have this strand that has separated from the other strand. So each original strand serves as a template on which the complement is built. So very important here, set each original strand. Sometimes when learners write, they will say both strands will act as templates. Then after that, when the strands have separated, then you get the free DNA nucleotides in the nucleoplasm that are going to build on these original strands to form new DNA molecules. Then when that happens, the complementary structure or complementary nitrogenous bases, your A is always connected to T or joined to T, which is adenine and thymine, and then your cytosine is always linked or joined to guanine. It's very important here. These are the pairs that are formed on your DNA molecule. Then this results in two DNA, or in two identical DNA molecules. Then at the end, each molecule, now the new molecule, will have the original strand and it will have the new strand. So this is important. These steps, the way that I've described them now, are as this when you look into your exam guideline. So when you are lost and you have these different resources in front of you that you are using to study, if you are unsure, especially for this process, refer to your exam guideline and just read those steps as they are written there. Then you will definitely get all the marks. You are not going to lose the marks. Then while we're looking at the DNA, there's also this topic that the examiners like very much, which is DNA profiling. With this one, you are not going to get straightforward questions that will be asking you maybe identify or name, but the questions that you'll get here are application type of questions. For example, we know that DNA profiling is used in paternity testing. It is used in identifying bodies and so on and so on. So what we have here, for example, when you look on this diagram, this is what we call the DNA profiles. What is given here are the DNA profiles of the suspect and the DNA profile or the DNA sample that was found in the crime scene. So what do? The question would be normally, this is the DNA that was found in the crime scene, and then here are the three suspects. Which one do you think is the suspect based on the DNA sample that is found on the crime scene? So what you need here is a ruler. Yes, you can just by looking at them and say number 1 or number 2 matches what is found on the crime scene. But I think it's better that you make use of your ruler, so that then you take and put your ruler next to the first band there, and then when we draw a line like this, we can already see that crime scene, the DNA on the crime scene and the DNA sample of suspect number 2, they have these two bands in common. Then if you move on to the next one, again, when we draw a line, remember when you'll be using your ruler, you put your ruler on your question paper and you look at the lines. If they match, then you know. So you can already see again here that the crime scene suspect, the DNA sample on the crime scene and the DNA sample of suspect number 2, they are also the same. Then by just looking, going down further, we can conclude that suspect number 2 or person number 2 is the suspect because their DNA sample matches with that DNA that was found on the crime scene. But now, how do you respond to this question? Avoid writing incomplete sentences. Remember your answer. You cannot just say the DNA of suspect number 2 matches the DNA of suspect number or matches with the DNA that was found on the crime scene. You are not going to get full marks. What is important is that you must mention that the bars or the bands on the DNA profile of suspect number 2 matches with the bars or the bands of the DNA profile that was found on the crime scene. So you mention your bars or you mention the bands and you mention the DNA profile. Remember, these are not lines. We are not talking about lines. We talk about the bars or the bands because these black bars, they represent the DNA fragments of a person. The nucleotides are the ones that appear as the dark bands as you see here on the DNA profile. Now, moving on. And then the other commonly asked question is protein synthesis. Even here, it's very rare that you will get a straightforward question where you will be asked to describe how translation takes place or how transcription takes place. But what you will get is the diagram, like the one that you see here, or half of this diagram that you see here. Then questions such as identify the processes maybe that are taking place at A and the process that is taking place at B. So by just looking at A, we can see that this line here represents the nuclear membrane, and there at the top we have the DNA. It is something is happening here. It has unwind, and the strands have separated. Then the clue, you have a clue here, the mRNA. So you know that already this falls under protein synthesis, and obviously the first phase that is taking place there is transcription. Then the second part of the diagram then represents translation. So again here, I said to you when I started, protein synthesis, transcription, and DNA replication are sometimes confused. So just pay attention and be careful of the things that I'm going to mention, because both of them, they are more or less the same. The first thing with transcription is that the DNA unwinds, which is the same thing that takes place in DNA replication. And we talk of the weak hydrogen bonds that break and the DNA strands, they unzip. Again, the order here, that's another common mistakes that you do. You cannot start by saying the DNA separates and then you come back and say the bonds break. Remember, it's the weak hydrogen bonds that break. Then because of that, then the strands are going to separate. After that, only one strand of the DNA acts as a template onto which the mRNA will be built. Remember, with the replication, we said each strand or both strands. Here, only one acts as a template. And how is it built? Built using the free RNA nucleotides. Do not leave that part out. With replication, it's the free DNA nucleotides. With transcription, it's the free RNA nucleotides from the nucleoplasm. And then the mRNA that complements the DNA in the nucleus will be formed. Then remember here that with the RNA, we said, the DNA replication, we said A always complements T and C complements what? Complements G. Here, yes, you will have C complementing G, but then A will be complemented by what? By U, because remember in the RNA there is no thymine, so this is not there. Remember how they complement each other. Then this first phase takes place in the nucleus. Then after the mRNA has been formed, there is our mRNA, it's going to leave the nucleus and it is going to go into the ribosomes, where the second part of the process is going to take place, and this is now called translation, and it takes place in the ribosomes. So here, what has happened is the DNA has provided a code. It provides the code on to how the protein will be built. So it carries this information that this is how hemoglobin, for example, which is a protein, should be built. So the mRNA takes that code from the DNA, leaves the nucleus, it goes to the ribosomes, where the process is going to take place. In the ribosomes, when it gets there, then the tRNA, now we have the tRNA. This is the tRNA. The tRNAs are going to bring the amino acids. When they do that, we look at here at the bottom we look at the base triplets. The base triplets of the DNA, we call them the anticodons, whereas the base triplets of the mRNA, they are called the codons. So when these two come together, the tRNA ensures that its anticodons matches the codons on the mRNA. And as that happens, as each tRNA brings amino acids and line them up against the information that has been provided by the mRNA, then these amino acids, they are joined together by the peptide bonds, and a protein is formed. For example, when you look into this, here we have ACA. This is the codon from the mRNA, and then this anticodon that is brought by the tRNA, or this amino acid with it, it's going to match with this, the UGU, because remember we said this is the pairing of the base triplets when transcription takes place. So here we have your A, that is the U that is going to pair with A, the C that is going to pair with G, and the U that is going to pair with A. Then we have the amino acid brought here, the first amino acid, and then the second amino acid, and then the third amino acid, and then together they are going to be joined by bonds and we'll have a protein at the end. So with this section, remember you will get a question like this, a diagram, or you get a table. Usually when you are given a table, you get a table with you are either going to be given the triplets of the DNA and the name of the amino acid, and then the diagram like this. Then you need to be able to work from the DNA to the mRNA and then move on to the tRNA. Sometimes you are asked to work backwards because you will be given the tRNA and you will be given the amino acids. Then the question will be asking you to identify what you think was the original DNA strand that provided these tRNA. So there you work backwards. So all the question papers that you are going to get, that you will gonna lay your hands on from today up until Sunday, make sure that you practice as many questions as you can. Try to work from the DNA to the tRNA, or work from the tRNA backwards. And then the other tip that I can give you, the DNA triplets, they are exactly the same as what you will get on the tRNA. But remember, the only difference is that the T is replaced with the U. So if you have your DNA and tRNA on the side, what is in the middle is going to be different, and that is what we call the mRNA. So DNA, the same as the tRNA, except for the T that is absent there. So this is protein synthesis, and this is a frequently asked question. Here are free marks that you can get on Monday. So moving on, then we've come to the end of eDNA. Remember, it is not only these things that are mentioned here. It is important that you know that when we talk about the RNA, there's three: there's messenger RNA, there's transfer RNA, there's ribosomal RNA. Remember the functions of the DNA and the functions of the RNA. You can be asked to tabulate between the DNA and the RNA. Importantly, be able to identify diagrams. When you read the questions, highlight the things that make sense to you. When you are given time to read, read with understanding. Then moving on to meiosis, which is 21 marks of the topic. So here with meiosis, remember this is the type of cell division where diploid cell undergoes 2 cell divisions to form 4 haploid cells. So sometimes when we describe this, we say it is the type of division that results in the formation of sex cells, or it results in the formation of gametes. Then important to know what happens before meiosis. What does the cell do to prepare itself for meiosis? And then we have the first division, which is we call meiosis 1. Then we have second division, which we call meiosis 2. And then what is important is the biological importance of meiosis. As said when I started, diagrams. Diagrams are very important. So here, questions that you might come across are, you will be given a diagram that represents a phase. Then the question would be, identify the phase that is presented in front of you and provide reasons. Remember, the reasons are the visible reasons, what you see in the diagram. You cannot provide reasons of anaphase that are not shown on the diagram because you know what those reasons are. It's visible reasons. So what you see in the diagram taking place, that will inform you what type of or which phase is being referred to in the diagram. So those are the questions that you will get. You can also be asked to draw the phase prior to the one that is presented in the paper, or the phase after the one that is presented in the paper. So those are very important. Or you might get a question that will say, how many chromosomes do you think this cell will have at the end of meiosis? So there, for example, you are given a prophase phase with 4 chromosomes. So now you must think how many chromosomes will be available at the end of division, and in that case there will be 2. Then the other thing that you must know, when you are given a diagram, it might represent a human cell, but only 4 chromosomes will be shown there. But in the statement, if it is mentioned that this is a human cell, and what do you think at the end the chromosome number will be? Remember that humans, how many chromosomes do we have in our somatic cells? There's 46. And at the end of cell division, meiosis, the gametes, each gamete has 23 chromosomes. So that's where we're coming in. So the terms that are important here, we are not going to go through all of them. Homologous chromosome, bivalent chromosome, chromatids. Remember, these are the ones that we normally confuse. So check the differences between the 2 chromosomes and chromatids. What is the centrosome? What is the centromere? Centromere is the structure that holds the 2 chromatids together. The centrosome is the structure that divides and gives rise to the centrioles, which then form spindle fibers. Then crossing over and chiasma. Remember, crossing over, it takes place in prophase 1 when the chromatids or when the chromosomes exchange genetic material. And the point where this process takes place is the chiasma. Then you have karyotype, non-disjunction, karyokinesis, cytokinesis, division of the cytoplasm here, and the one at the top, division of the nucleus. Then somatic cells, sex cells, autosomes, gonosomes, random arrangement, it's very important, diploid and haploid. So these are the terms that you must know. From the beginning of the year, you were taught the strategies of learning the terms. You are given the terms, you give the terminology, or you are given the term and you are asked to describe what this term means. Then the process of meiosis. We said it starts when the cell prepares itself for division. the first phase or the preparation phase, it's what we call interphase. Here, this is where DNA replication takes place. So it takes place just before meiosis can resume. And in this phase, what is going to happen is that chromosomes will now consist of two chromatids that are joined by a centromere. Remember, we have chromatin. Before everything takes place, there's chromatin network in the nucleus. And then those, they shrink, they become visible chromatids, and during that whole process, DNA replication takes place, and then we have visible chromosomes. So we said with the DNA replication, it helps to double the genetic material so that it is shared equally amongst the new cells that are formed at the end. Then hence here we talk of the unreplicated chromosomes and replicated chromosomes. The unreplicated chromosome, it is a single chromosome as it appears before cell division or before DNA replication. Let me just go to them so that you can see what I'm talking about. This shows the replicated and the unreplicated chromosome. So a chromosome that is unreplicated looks like this. This is before DNA replication. And then when it's replicated, it is when you can clearly see the two chromatids that are held together by the centromere at the center there. So we have the replicated and the unreplicated. This is before interphase or while interphase is taking place, and then at the end of interphase, just before then cell division or prophase 1 takes place, it becomes replicated, and this is what you see. So the differences between the replicated and unreplicated. Then how do we then identify? I said the questions that you will get, you will be asked, you will be given the phase, and then the question would be, what is this phase? How do you know that it is this phase? So these are the tips that you can have a look at. With prophase 1, how would you know that it's prophase 1? First of all, you will see that crossing over is taking place. The homologous chromosomes will lie next to each other, and when they do that, they then exchange genetic material. Here is crossing over taking place, and the point where it is taking place, we said it's called the chiasma, and then at the end, they will look like this. So now we say homologous chromosomes, they lie next to each other. During this stage, when they are exchanging genetic material, when crossing over takes place, this is the only time that we call them the bivalent. They are in this form during this time. So to avoid making use of bivalent when you see it in other phases, you can only identify homologous chromosomes as bivalents only when they are exchanging genetic material during prophase 1, when crossing over is taking place. So those are the things that you will see. Another tip, you will see that the nucleoplasm or the nucleolus or the nuclear membrane, it disintegrates or it disappears, and the spindle fibers start to form. The centromere, I said the centromere is the structure that gives rise to the centriole. So the centromere will then give rise to the centriole, which will split when they split like this, and then you see the spindle fibers form. Important, crossing over leads to variation, because we're saying these new cells that are formed at the end, they are not genetically the same because of variation. And then prophase 1, on the side, there is no crossing over that is taking place. That's what you can see. But the nuclear membrane again and the nucleolus are disintegrating or disappearing, but there is no crossing over. Then when you go to metaphase, I think this is the easiest to identify, because all that you look for the chromosomes that are lying along the equator of the cell. Remember, avoid using in the middle, avoid using at the center. They are lying at the equator of the cell, the equator here. That's where the chromosomes are. So with metaphase 1, they are lying in pairs. So you'll see two lines of the chromosomes. Whereas in metaphase 2, chromosomes are lying in one line, singly, like this. They are not in pairs. So that is how you identify metaphase 1, metaphase 2. So with metaphase 1, the centrioles are already at the poles, and then chromosomes come lie on along the equator. Again, remember, they are pulled by these spindle fibers. They are the ones that are pulling them towards the equator of the cells. But this process does not depend on anything. This moving towards the equator of the cells happens randomly. We talk about the random arrangement of chromosomes. It is not informed by any pattern. They are just drawn by the spindle fibers, pulls them towards the center anyhow. So because of that, that is why we say it leads to what? To variation. Because if I have two cells next to each other and metaphase is taking place, the way the chromosomes are arranged on cell number 1 will definitely not be the same as the way the chromosomes are arranged on cell number 2. Okay, I don't know what happened there. Okay, I'm trying to just go back. Give me a minute. Oh. Okay, can't go back to. I just use my. Just quickly gonna go back to the original slide. Yes, this is where we are. So they are there at the equator of the cell without any pattern. So I was saying, if you have two cells next to each other, what happens in metaphase, the way the chromosomes find themselves along the equator will not be the same, because there's no process, there's no pattern that they follow. Then the differences between the two is this. Again, in metaphase 2, random arrangement takes place, but the random arrangement here, it's now the chromosomes are forming a single line, whereas on the other side, they're still lying in pairs, but which are not informed by any pattern. Then we have, after that, the next phase is anaphase. Here, then the chromatids now start pulling the homologous chromosomes from the equator towards the poles. Chromosomes move towards the poles. They are being separated from each other on the anaphase. That's how you identify. So the spindle fibers, we say the spindle fibers, they contract. When they contract, then they pull the whole set of the chromosome towards the pole, and this side, these ones are pulled towards this pole. On anaphase 2, remember I said they form a line. So here in anaphase 2, the centromere splits. When it splits, then each chromatid is pulled towards the pole. Because remember, a chromosome is made up of two chromatids that are held together by a centromere. So in anaphase 2, the centromere splits, and then these chromatids then move towards the poles. Then we'll talk about this in the later stage. But then if this does not happen, if it happens that the whole set is not separated and it goes towards the pole like this, then we talk about non-disjunction, where the homologous chromosomes fail to separate and two of them move to one pole. Then it means the new cell the cells that will be formed will have one cell that does not have chromosomes in it, or will have a cell that will have extra chromosome in it. That's when we talk of non-disjunction. And then in humans, if we have that extra chromosome on chromosome 21, we talk about Down syndrome. So the opposite is on the other side. And then the last phase is telophase. With telophase, then that's when now the nuclear membrane reforms. Remember we said at the beginning, the nuclear membrane disintegrates or it disappears. The chromosomes become visible and crossing over takes place. At the end of cell division, or at the end of telophase 1, the nucleolus or the nuclear membrane reappears, and the cell membrane constricts. As you can see there, it is constricting, and cytokinesis take place, which is the division of the cytoplasm. And then we will end up with two cells. So these are the two cells. Then we'll continue with meiosis 2. what we saw when we started, because the phase are next to each other, so that we make it easier for you that you can see the differences between meiosis 1 and meiosis 2. And then at the end of telophase 2, there's 4 cells, because remember when we started with 1 cell, and then at the end it gives rise to 2 at the end of meiosis 1. Then those 2 cells continue with meiosis 2, and then at the end of meiosis 2, there will be 4 cells. So it's the same thing. At the end of telophase 2, you have single-stranded or unreplicated chromosomes. Then each daughter cell has half the number of chromosomes, and we have 4 daughter cells that are genetically different. And this genetic difference has been contributed to by your crossing over in prophase 1 and random arrangement in random arrangement in metaphase 1 and metaphase 2. Then non-disjunction, mentioned non-disjunction during anaphase. We said here, this happens when the homologous chromosomes fail to separate. So when the homologous chromosomes fail to separate, it means they move as a pair towards the pole, and then we might end up with a cell that has extra chromosome, or we might end up with a new cell that does not have chromosomes. So in human cells, when you talk about this, usually it happens on chromosome number 21. Chromosome pair number 21, when they don't separate, it means that the new gamete has an extra copy of the chromosome. So when this gamete with 24 chromosomes fuses with a gamete with 23 chromosomes, then we say it may lead to Down syndrome. So this is the only disorder that we're supposed to know in grade 12 as it appears in our exam guideline. Down syndrome, which is caused by non-disjunction, failure of chromosomes to separate. So this will either happen in anaphase 1 or in anaphase 2. So in anaphase 2, we're saying the centromere will fail to separate, therefore the whole chromosome will move to the pole. Then here again, you will get questions where diagrams will be given, have to identify, and then if you see, or you'll be given the human karyotype, how the chromosomes are arranged in a cell, then you can just go and look at chromosome number 21. If you see that there's an extra number there, then you know that Down syndrome or non-disjunction has taken place. Then at the end of this, just to wrap up meiosis, you might get questions, although it's not on the exam guideline, you might be asked to differentiate between mitosis and meiosis. You might be asked to differentiate between meiosis 1 and meiosis 2. And like I said earlier, you might be asked to differentiate between RNA and DNA. So we've come to the end of the first session of DNA and RNA. What I'd just like to remind you is that, remember, for your revision, especially meiosis you will find in paper 1 of the previous papers. It's the only topic that appeared in both papers, in the old papers. But as from this year onwards, it is only found in paper 2. So we're going to take a 2 minutes break, and I will then allow Redin to continue with genetics.
Hello grade 12s, my name is Radine Kruger and I'm going to be covering the topics genetics and inheritance and evolution through natural selection with you. So let's have a look at the first topic, genetics and inheritance. And as you can see, it's for 48 marks of the exam paper, which is almost a third. So this section is very important. Okay, so there are quite a few subtopics in genetics and inheritance, and they are listed here on this mind map. So if you have a look, we can start off with terminology, then we look at monohybrid crossing, sex determination, sex-linked inheritance, dihybrid cross, blood groups, genetic lineages or pedigree diagrams, genetic engineering, paternity tests, genetic links, and Mendel's principles of inheritance. Okay, but today we're going to focus on the concept of inheritance. We're going to have a look at some of the terminology. We're also going to have a look at Mendel's laws. Then we're going to have a look at monohybrid crosses and the different types of inheritance that we get. Then we'll look at sex-linked inheritance. So we have a look at, how those are inherited. We'll look at family pedigree diagrams. And then how certain characteristics are inherited in families. We're not going to look at genetic engineering, but what is important is that you do know these concepts. So you know what stem cell research is, you know what genetically modified organisms or GMOs, as they are commonly spoken about, and that you also know the process of cloning. Right, and then we'll quickly briefly look at paternity testing. Right, the terminology that we find that is commonly confused and sometimes used interchangeably are the following. So the first one is chromatin and chromosomes. So we know that in the nucleus of a cell, there's chromatin network. That is what we call chromatin. So it's a long strand tangled in the cell, and you can't actually see anything distinct. However, as soon as cell division begins, whether it be mitosis or meiosis, the DNA then condenses and it forms individual chromosomes. So the chromosomes then are just a different stage of chromatin network, and chromosomes are visible during cell division. Right, then the next one, and this one's very important, and I know a lot of learners mix them up, and those are genes and alleles. Now, you know that there are genes, and the genes are coded on the DNA, and they are responsible for certain characteristics or proteins or whatever you'd like to call them. Now, those genes can have different forms, and in most characteristics, you find two forms of a gene. So, for example, let's look at eye color. So we get a gene for, or an allele for brown eyes, and you get an allele for blue eyes. So although it's the same gene, which is for eye color, there are two different forms, and those two forms is what we call an allele. Right, then we have a look at the difference between a dominant allele and a recessive allele. So the dominant allele usually is an allele or a form of the gene that dominates or is displayed in the phenotype of the individual. So, and that usually masks the phenotype of the recessive allele. So if as an individual you have one dominant allele and one recessive allele, then you will have the physical characteristics of the dominant allele. Okay, so the recessive allele usually is masked. So to see the phenotype or to have the phenotype of the recessive allele, an individual needs to have two recessive alleles. Right, that was dominant allele and recessive allele. Right, then the next terms, or the next two terms that sometimes are mixed up, and this is especially in a cross, where you write down phenotype and then you actually give the genotype, and then you write down genotype and you actually give the phenotype. Now the difference is pheno is the same as physical, all right? So that The phenotype is the physical characteristic of the individual, whereas genotype is the genetic makeup. Okay, so remember, pheno for physical and geno for genetic. Okay, then if we look at the next two terms, we see the first one says homozygous and the next one says heterozygous. Now the two stems, or the stems of those two words, homo we know means the same, whereas hetero means different. So when we talk about a homozygous individual, we mean that both the alleles for a gene are the same. Whereas in a heterozygous individual, we find that the two alleles are different. All right, so that's the terminology. Please, if you're not sure, please have a look in your textbook or in Mind the Gap to make sure that you understand the difference. Right, then let's look at Mendel's laws of inheritance. There are 3 laws. They are stated in the exam guidelines clearly. So the first one is his law of segregation. And this one is the most frequently asked. The second law is the law of dominance. And his third law is the law of independent assortment. So in other words, alleles of different genes assort independently of each other. So in other words, if you get the eye color from your father, you may not get the shape of eye from your father as well. You may get your mother's shape of eye. So it means that genes separate independently of each other. Right. Okay, now the format of a genetic cross. And here these questions usually are worth 6 marks. And out of those 6 marks, you virtually can get 50% of those marks, which is 3. And that is because there's a recipe to writing out a genetics cross. So you can either do it like this, where you do that kind of cross, or you can do it in a Punnett square. Remember, to start off with, we write P1 and F1, and they should be in that position. So P1 indicates the first parental generation, and F1 represents the first filial, that's what the F stands for, or offspring, so that generation. Then we write out phenotype, and here that is usually, as we said, was the physical, the visible traits is the phenotype. So for example, tall plants or short plants. Then we write out genotype, and that is, as I said, the genetic makeup of the individual. For example, we might have 2 big T's, so that individual is homozygous, or big T, small t, or 2 small t's. Then we write out meiosis. So meiosis comes in next, because from meiosis we form gametes. Okay, and then the gametes, when we write out the gametes, so say we're using the genotype, this is the genotype, then what we need to do is we need to separate out the gametes. So there should be a space, or I'm going to put a semicolon between the 2, or the 4 of them, if you write all 4 of them out, just to indicate that they are gametes and not a genotype. Okay, and here I would put out big T, small t. Right, so once the gametes have been formed, to get offspring, fertilization must take place. So if I put those 2 in the correct place, I'm going to get a mark for meiosis and fertilization, and I'm going to get a mark for P1 and F1. And that is what I meant when I said you're going to get an easy 2 marks. The third place that you can get a free mark is usually in the question. They'll tell you a tall plant was crossed with a short plant. So I'm going to write the phenotype there. So I'm going to say tall plant and short plant. Okay, so there I've already got 3 marks out of 6 without actually doing very much. All right, so once we've worked out the gametes, then we're going to cross them, and we'll get the genotype of F1, and we write that down. So that is then going to be fertilization taking place. And then from our genotype of F1, we can then write down the phenotype. All right, the second way of writing out your genetics cross can be, you can use a Punnett square. So this little diagram here in the middle, we call that a Punnett square. And again, let's use the same genotype. And what we do is we place the gametes into our Punnett square. So we can put the big T and a big T, and we place this big T and a small t. So it goes in the first row in the first column, and then we're going to actually carry out fertilization, and we are going to get the genotype of our individuals. So if we have a look, this is what is happening. Okay, so there we've now worked out the genotype. Remember, once again, we've written P1, F1, we've written meiosis and fertilization, and they should be in the correct position. Okay, so remember, after genotype, meiosis is going to occur to form the gametes. Once we have the gametes, to get the offspring, fertilization must take place. Okay, so that is where we position them. So remember, there's your free marks, and we're always looking for free marks. Okay, that ensures that we either pass or we're getting our distinction or whatever we are going for. Okay, so, look at how you set out the genetics cross. Right, then we're going to have a look at the types of inheritance. And if we have a look at the first one, monohybrid, remember mono equals 1. So in that kind of cross, we're going to only look at a single characteristic, at one trait. And then we have a look at complete dominance. So here we have where one characteristic is dominant over the other. And we look at incomplete dominance, and we see that neither of the characteristics is dominant. In codominance, we're going to see that two characteristics will be equally dominant. Then we're going to have a look at sex-linked disorders, and then at a dihybrid cross. And remember, di equals 2. So it's important to know the difference between monohybrid and dihybrid. You won't be given a dihybrid cross to do in the exam, but you could be given the results of a dihybrid cross, and they can then ask you questions on it, and maybe even ask you what type of cross it is. So remember, when we're looking at two characteristics, it's a dihybrid cross. When we're looking at one characteristic, it's a monohybrid cross. Right, so true complete dominance is when we have two homozygous individuals as parents, and when you cross them, can you see the one parent has the dominant alleles and the other parent has the recessive alleles. So in the first generation, in the F1, we're going to find all our offspring are heterozygous, but because they have the dominant allele, they will then all have the phenotype or the physical characteristic of the one parent. So then only one of the parents' phenotype is visible in the offspring. So that's how you remember complete dominance. Only one parent's phenotype is visible in the offspring. Right, then we look at the second type of inheritance, and that is incomplete dominance. And here we have two different types of flower or colors of flower. We're looking at the flowers. And if we have a look at the offspring, so they would be P1. And if we have a look at the offspring, this is F1. And we can see that they are different. They don't look the same as the parents. So we have a red flower and a white flower. When we cross them, we have pink flowers. So if I give you a pink flower and I say to you, what did the parents look like? You wouldn't be able to tell me. You'd have to first cross those individuals to find out. And that is because it is incomplete dominance. Right, so remember, in incomplete dominance, we see neither of the parents' phenotypes in the offspring. Right, then we go to the next type of dominance. It's called codominance. Co means when we're working together, or there's two that are the same. And in this case, we see, for example, one of the examples is in cattle, where we have a white cow and a red bull, and we cross those two, and we find out that we have a offspring. It looks like a cow, that has got both white and red hair. Okay, so when we have a look at those offspring in codominance, we can actually see both the parents' phenotype. Okay, so it's very easy to remember the different types of inheritance. In dominant inheritance or in complete dominance, we have only one parent's phenotype visible or evident in the offspring. In incomplete dominance, we have none of the parents' phenotype in the first generation. And in codominance, we see that both parents' phenotype is evident in the offspring. Right, then we look at sex determination. And I hope you all know what this sign means, that symbol. That is the symbol for males. And basically what we have here is a male karyotype. It's what we call a karyotype because it shows us all the chromosomes of a male individual. Okay, so if we have a look at the first 22 pairs of chromosomes, those are all known as the autosomes. Okay, so you need to know what the term autosomes mean. And then if we have a look at the very last pair of chromosomes, we see that they are slightly different because this is a male, and in fact they are an X and a Y chromosome, and those then are known as our gonosomes. So the gonosomes or the sex chromosomes are the last pair, and the first 22 pairs are the autosomes. Right, then this, now obviously, and it's nice and pink, happens to be the symbol for females, and this then again is a karyotype, which indicates the type or the chromosomes or the structure of the chromosomes of a female individual. And again, the first 22 pairs of chromosomes are known as the autosomes, and the last pair again is the gonosomes. And in this case, you can see both of those chromosomes are the same, which means then that the individual is a female. Okay, so I'm going to go back again and just show you the difference in a male karyotype. Can you see here the difference in the size of the chromosomes? So there we can see one large chromosome, which is the X, and then the smaller chromosome is the Y. So if you are given a karyotype in the exam, you would be able to identify it by having a look at the last pair of chromosomes, because that will then indicate if there's two of the same chromosomes or two different chromosomes. If it's two different, it will be male. If it's two of the same, it will be female. Okay, so that's how we determine sex. And then when we do a genetics cross, we work out, we can see the mother has two X chromosomes. Mother has two X chromosomes, the father has an X and a Y. And remember, when meiosis occurs, they separate out, and the mother then can give all her offspring the X. So she will give the X to all her offspring, as you can see there. And it's actually the father that determines the sex of the child, because he will give one X or an X to two of his offspring, and the Y will go to the other two. And as soon as an individual has an X and a Y, they are male. If an individual has an X and an X, then they are female. So there's a 50% chance of having a boy and a 50% chance of having a girl. And it doesn't matter whether the examiners ask, because they love to do this. This is their favorite question. Mr. and Mrs. Ndlovu have three children, 3 boys. What is the chance of them having a daughter as a 4th child? It's not, don't think of it as 25% because it's the 4th child. There will always be a 50% chance. Because remember, the father can either give an X chromosome or he can give a Y. Okay, so there's a 50% chance of them having a daughter. Right, then we're going to look at those disorders that are linked to the sex chromosomes or the gonosomes. So these are genes that we find that are located. So if we have a look, genes that are on the sex chromosomes, and they are called sex-linked, not sex-linked, sorry, sex-linked genes. And usually when we look at the sex-linked disorders in humans, we usually refer to those that are on the X chromosome. So we call them X-linked. Right, and that means that these genes only occur on the X chromosome. And that is very often because if you have a look at the size of the Y chromosome, there often isn't space on the chromosome for some of the genes. And that is why they are then only found on the X chromosome. All right. So fathers can pass on the X-linked allele to their daughters, obviously, because remember, if the father gives an X, the mother can only give an X. Then they're going to get an X, and that will be a daughter. So they can't pass on an X-linked disorder to their sons. However, mothers will pass on that sex-linked allele to both their sons and daughters. Okay, well, there's the possibility. Right, the first disorder, and this is in the exam guideline, you should know, hemophilia. And we see that here in this cross, okay, the father does not have hemophilia, whereas the mother, she's normal. Okay, we don't call her a carrier. She's normal, but she does carry the hemophilic gene. Okay, so there you can see she has it. So when we have a look at the cross, when it's passed on, you can see the mother has passed on that X to two of her, because remember, two of her offspring, and the other two offspring received the normal X, the allele for normal blood clotting. Okay, so the father can only pass the affected allele to his daughters, and in this case the father does not have the allele, but the mother can pass on to both her sons and her daughters. Right, then the second X-linked or sex-linked disorder that we're going to have a look at is color blindness. Now, if we have a look at this picture here on the right-hand side, you can see that the first example shows normal vision. Okay, so I can see the cap is green, the t-shirt is yellow, and the tackies are red. However, if I have red-green color blindness, then I confuse the red and the green. I don't swap them around. You can see that individual can't see red and green. What they see is different shades of yellow. Okay, and that is what happens with someone who is colorblind. Now again, as we said, it's an X-linked gene. We have a normal allele, and you can see it on this chromosome, and then we have the allele for color blindness on this allele. Okay, remember these are each X chromosomes. The father, he's normal. He doesn't have color blindness, so he has the normal allele, and on the Y chromosome there's no space for that allele, so he just doesn't have an allele for vision. The possible offspring, so as fertilization occurs, we see the outcome, and we see one of the females will be normal. The other female is normal, but she carries the allele for color blindness. So she can pass that on to her sons, and in fact her daughters too. The next individual is a normal male, and the second one is a colorblind male. So he's not even a carrier. He has color blindness because he has the allele for color blindness, and there is no other allele to mask that characteristic. Okay, so that is how our two sex-linked disorders are inherited. In the exam, if they give you another example, then they will give you all the information that is required. But these ones, you need to learn them. You need to know that it's caused by a recessive allele and that the allele is on the X chromosome. All right, now we're going to have a look at blood groups. And here we have a look, and we can see that inheritance of blood groups is due to multiple alleles. Okay, remember at the beginning when I was explaining the difference between a gene and allele, and I said to you most genes have two alleles or two forms of that gene. Now in blood groups we don't have two forms of the gene, we actually have three. Okay, so the notation or the symbols we use for these alleles is an I with an A, an I with a B, and a small I, okay, which indicates blood type O. Right, so I'll have we'll have a look at them now a little bit further on, but we can see there's different different combinations, okay, of those alleles, and it's those different combinations that result in the different 4 blood groups. So here in this diagram here you can see, and in fact this is what we call a pie chart. You guys will be familiar with that, and I hope you know how to draw them. But in this pie chart you can see the amount of the population that have blood type O, which is the most, blood type A, which is slightly less, but still a large portion of the population. Blood type B is a smaller portion, and blood type AB is the smallest portion of the population. Right, so when we, actually let me first go back, sorry, before I get there. So now when we have a look at the blood groups, and remember blood group is the phenotype. Okay, that's the physical characteristic of the individual. I have blood group A, you have blood group B, whatever. Okay, so that is the physical characteristic. Then when we look at the alleles for that blood type, that's when we're looking at the actual genes. And this is where we're then going to use the notation that I spoke of a little in point 2. Now in blood type A, an individual can either have an I with a capital letter A and a second I with a capital letter A, as you can see here, and that we called a homozygous individual. Remember, these 2 alleles are identical. Or they could be heterozygous for blood type A, and that means they have the allele for blood group A as well as the allele for blood group O. And in this type of inheritance, that would be an example of complete dominance, because we've said the individual has blood type A, yet they have a different allele, but that allele is recessive. So in this case, it is complete dominance. If we have a look at B, blood type B, again we have a homozygous individual that has 2 alleles for blood type B. Then we have a look at the second possibility is a heterozygous individual which would have the allele for blood type B as well as the allele for blood type O. And again, that type of inheritance is complete dominance. The next example, this is blood type AB. And here, if we have a look at the alleles of the individual, we see that both allele for blood type A and the allele for blood type B is present in the genotype. And remember, when we spoke about codominance, we said both the parents' phenotypes would be visible in the offspring. So in this case, we have both the alleles of both the parents, and the individual then has blood type AB. And that is an example of codominance. Right, then in the last example here, blood type O, we see that there is again a homozygous individual, and as we said, the allele for blood type O is recessive, and therefore this individual has 2 recessive alleles, and therefore would be blood type O. And again, that would be complete dominance. All right, now let's have a look at genetic lineages, or what we commonly know as a pedigree diagram. And please understand the difference between a pedigree diagram and a phylogenetic tree, which we will discuss later on in the program. Okay, so a pedigree diagram is a genetic inheritance where we can see the characteristic as it is passed on or inherited in a family. So we usually have more than one generation, and we can then trace how the characteristic, whether it be a disorder or any kind of characteristic, how it is passed on from one generation to the next. Now, very important when we are faced with a pedigree diagram, we need to understand the symbols that are used. So if we have a look, and this you should know. Okay, so we know that a circle, this is a female, a square is a male. As soon as the square or the circle are colored in, like this one, so as soon as they are shaded or colored in, that then becomes an affected individual. So they would have that characteristic that we are tracing through the generations. We don't give deceased individuals. We don't usually use that. We don't often use twins. We also don't use adopted and miscarriage. So the first three are very important here. These ones you need to know. Then, how are they joined? How are those shapes joined? If we have a look at this line over here, we see as soon as it's a horizontal line between two individuals, that indicates a marriage line. A vertical line going down like this is usually our line of descent. And that would then indicate that this next generation is the offspring. Okay, and here, so, and then a line joining individuals in that next generation are then known as siblings. So in other words, those would be brothers and sisters. Also, what we can see on this pedigree diagram is the different generations. So if the examiners ask you how many generations in this diagram here, and you would be able to say that there are 2 generations. Or they would say to you, in the first generation, which individual? And then you would go and look at the very first one. Okay, so it's important that you know the structure of a pedigree diagram. Right, now the steps in solving a pedigree diagram. Okay, so the first thing we need to do is we need to look at the key. Okay, and that's the key that they give you that says this is a male, this is a female, this is an affected male, this is an affected female. Then we go and look for the dominant and the recessive characteristics. Okay, once we've figured out which is dominant and which is recessive, and they will usually give you that in the stem, then you fill in the genotype of the individuals with the recessive condition. So if we have a look at this diagram here, at the bottom they're giving us the symbol or the key for the recessive individuals. So I would go and have a look and I see these are all recessive. Okay, so I identify them first. And then remember we said as soon as an individual is recessive, they must have 2 alleles that are the same. They are homozygous. So then we can very easily fill in the recessive or the alleles for those, or the genotype for those individuals. So we work backwards and we find that there's 1 recessive allele from each parent. So we fill in those. Then if we want to work out for the rest of the individuals, then we have a look and we see if the parent has the dominant characteristic, then we're going to fill in 1 capital letter for the individual and then a second letter. All right, so we fill in the recessive characteristic and then we fill in the dominant characteristic. Any other individuals that show the dominant characteristic then are most likely homozygous dominant, which means they have 2 dominant alleles. Right, then the last aspect we're going to have a look at in genetics and inheritance is paternity testing, and this can sometimes, and it is in the recent past, has been asked frequently. So there's 2 ways in which we can do paternity testing. The first one is blood groups, where we can have a look at the blood group of the mother, the father, and the child, or the prospective father, because that's what we're trying to determine whether an individual is the father or not. So we have a look at their blood groups, and then we do a cross to see if we cross those 2 parents, would we get the offspring or the blood type of the offspring that is in question, and in that way we can determine whether he is the possible father. Now remember, in blood groups, there's only 4 different blood groups. So there are many individuals that have the same blood type. So actually when we do we do paternity testing through blood types, we actually really eliminate individuals that are not the father. It's not an absolutely definitive method of determining parent or fatherhood. The second method we use is what you guys saw earlier, is called DNA profiling. And there what we do is usually we have to analyze both the mother and the prospective father, or however many prospective fathers there are. We analyze their DNA as well as the child's DNA. Then we have a look at the profile, which is a pattern of bars. and we eliminate all those alleles or genes that bars that are common from between the mother and the child, as you can see here. And then what's left must match all of those, all the bars that are left must match one of the fathers. And if you have a look at the very first bar, we can see father 1 doesn't have that characteristic, but father 2 does. We look at the second characteristic, we see father 1 also doesn't have that characteristic, but father 2 does. So already we can see more than likely father 2 is the father. Then we go further down and we have a look at other common characteristics, and we see that father 2 has all. Can't be some, because a child only gets its DNA from its mother and a father. It doesn't get it from two fathers. So all the bars of an individual must match what is left on the child's DNA profile that is not common with the mother. Okay. All right. The next topic we're going to have a look at is evolution. Now, evolution is divided into, makes up 54 marks of paper 2, which again is more than a third, just slightly more than a third. So you can see that genetics and evolution make up the majority of paper 2. All right. Now, specifically, what to focus on. We're going to have a look briefly at the evidence for evolution, and it's very important that you do cover, or you do understand the evidence, because in the last couple of years they've been asking about the evidence for evolution quite frequently in the exam. Right, then we're going to have a look at sources of variation, and remember we spoke about variation in genetics. And then we're going to look at the theories of Lamarck and Darwin. Then we'll have a look briefly at punctuated equilibrium. We're going to look at artificial selection, speciation, and then Klantler will carry on with human evolution with you. Okay. Right, so let's start off with defining the concept. What is evolution? Okay, and it's important that we understand what evolution is, and it's a process of change, and the change occurs in the genes. Okay, that's very important. It's mutations and changes in the genes that drive evolution. So those genes have transformed life on Earth from its earliest forms to what it is known, or to what we know life as today. Okay, so biological evolution means that all present day forms of life, so everything we see, whether it be a unicell organism up to a human or a mammal, or, those are complex organisms. All of those have descended from and are related to organisms that lived in the past. Okay, so they may look different, and that is because in each generation, possibly, mutations occur randomly and they change the genes. So they may look different, and that is because they have become modified from one generation to the other. So if we have a look at the diagram here, we can see the ancestral animal that was the common ancestor of the horse that we know today. And you can see through the ages, as evolution has taken place, there are significant changes that have occurred. And so we know this as the modern day horse, but this would be the ancestor that evolved, or that the horse evolved from. Right, now the evidence. And there's 4 lines of evidence that you need to know. And you don't need to know it in great detail, but you just need to know how it is evidence of evolution. So the first one we have a look at is called biogeography, and that is kind of linked to your grade 10 syllabus, where you learnt about continental drift. So that is where the continents broke up. So if we have a look at, for example, the flightless birds here, we can see there's the rhea, South America, the ostrich that is common in Africa, which I'm sure you all are familiar with. We find the emu in Australia, the kiwi in New Zealand, and the cassowary in Papua New Guinea. Now, all of those are flightless birds, and they have characteristics in common. But how is that? Because they are flightless, they cannot fly. So how did we get similar organisms on all the continents that are separated by vast oceans? And that is evidence that at one stage all those continents must have been joined in a supercontinent, and so the common ancestor of all of those individuals originated on that large continent. Right, then the second form of evidence is known as genetics, and I mean we've spoken about it, and here we can compare, and this is where we do DNA profiling. We can have a look at mitochondrial DNA, and then we compare the similarities and the differences. And by looking at the similarities and the differences, we can work out how closely related organisms are. Right, then the third form of genetic evidence is known as homologous structures. And here we see, if we have a look at all of these individuals or these organisms, one is a human, a cat, a whale, and a bat. But if we have a look at the internal structure, we can actually see that the bone structure of all of these animals is exactly the same. The shape of each bone might be different, okay, but all of them have what we call the pentadactyl limb. So in other words, it's a limb with the 5 digits. All right, so, and the reason why they are different is because each organism has different uses for them, but it is evidence of a common ancestor. And then the last form of evidence that we're going to look at is the fossil record. And here there's a huge, a vast source of fossils that we have that have already been found, and they are then, we look at those and we use that as evidence as well. All right, then we need to look at the sources of variation, and this is a little bit of revision because a lot of this you have covered in earlier topics during the year. So the first source would be a mutation. And remember, a mutation occurs when DNA is replicating. So, and that a mutation, you know, is a sudden, unexpected change in the genetic structure of the DNA of a cell. All right, and if we have a look at this one here, we can see. So in this strand here, in this sequence of the DNA, we have an A, and in this one, we can see when the replication took place, it was misread and it was replaced by a cytosine. All right, so we have the adenine that has been replaced by cytosine, and as soon as we change the codon, you know that from DNA and RNA, we change the protein or the amino acid that has been or that is coded for. And when we have a change in the amino acid, that the sequence, then it will change the type of protein, and it depends on whether it's a lethal change, mutation. So in other words, we have a malfunctioning protein, or if it's a beneficial change, or if it doesn't make any difference. So, but mutations then create variation. Right, then the second source of variation comes in meiosis, and we've also covered meiosis this afternoon. So there you can see the first process that creates genetic variation is called crossing over. And as you know, your homologous chromosomes, okay, so one is paternal, one's from the father, and one is from the mother. And what happens is during prophase 1, the chromatids, or one of the chromatids of each homologous pair, actually physically cross over. Now remember, we learned this, and we learned that at the point at which it crosses over is the chiasma, and once the crossing over has taken place, we see that this chromatid now is not purely paternal anymore. There is a bit of maternal DNA there, and on this side we see there's a little bit of paternal DNA. So now if we have a look at the individual chromosomes, we can see each one is different. Okay, so that has created our genetic variation. The second source during meiosis is during random arrangement. And please, it's only random arrangement. So it's during metaphase. So it's here in metaphase where the chromosomes line up. Can either be in metaphase 1, which is indicated in this diagram, or it can even be in metaphase 2. And the whole reason why it causes genetic variation is because if you have a look here, you can see in this diagram all the red chromosomes on one side and all the green ones on that side. But another possibility, the next time a meiosis occurs, we can have 2 red ones and 1 green one, and 2 green ones and 1 red one on that side. And here we have them shared out. So we have 1 green, 1 red, 1 green. And here is a different form. So can you see once the gametes are formed, every single gamete will be different. And that is because of genetic variation. Right, then another form or another source, not form, another source of variation is reproduction. And the first thing is random fertilization, because remember, males produce millions and millions of sperm cells. So when intercourse occurs, the male would deposit all those many sperm cells into the female's body, and those sperm cells then make their way to the ovum. And remember, in humans, we usually only have a single ovum. Okay, but we don't know which one of those sperm cells is going to fuse or fertilize the ovum. So that once again, it's random fertilization. And the second form of or source of variation in reproduction is called random mating. And that is when this individual is able to mate with any one of these 3. Okay, so that is then random mating. So those are the sources of variation in reproduction. Right, now the types of variation. And these 2 concepts you need to know because they do ask them. The first one is called continuous variation. And that is when we look at characteristics of organisms where there's a range of intermediate phenotypes. So you can't categorize individuals into tall and short. Okay, there's a whole range of height. So if you have a look at this picture here of all these little girls, you can see there they all have different heights. Another example of continuous variation is skin color. Okay, we have various ranges of skin color. We also have various ranges of hair color. Okay, so none of these characteristics can be categorized into, 2 or 3 different categories. The next type of variation is called discontinuous variation. And here we don't have continuous or intermediate phenotypes. Here we have distinct characteristics. So they fall into a particular category. And a good example, or 2 good examples here, is blood type. We said you can either have one of the 4 blood types, and then also fingerprints. That's another very good example. Every individual has a unique set of fingerprints. All right, then we're going to go on to the theories of evolution. We're going to have a look at Lamarck, and he worked during the early 1800s. And basically, he had 2 laws that he used to explain evolution. And the first law was known as the law of use and disuse. The second law was known as the inheritance of acquired characteristics. Now, you need to know these laws. They are often either asked or you need to explain a particular scenario using these 2 laws. Now, if we have a look at the first, when we answer a question on Lamarck, we see that the law of use and disuse actually he explained it as an organism either using a structure and therefore it would develop, or otherwise not using a structure and then it would disappear. And then that, once it either has strengthened or it's become bigger or whatever it's changed, however it's changed, that is known as the acquired characteristic. And that acquired characteristic then is passed on, according to Lamarck, to the offspring. Okay, so when you get asked a query or a question on Lamarck's theory, the important thing is to determine whether the inherited characteristic is that of an offspring that was born with it, or is it an acquired characteristic? Is the scenario giving you acquired characteristic where they say the individuals develop, so they're not born with it? So the guiding question when we look at Lamarck's theory is we have to look at what was the original characteristic. Okay, so we need to determine that. Then we need to know what was the challenge. In other words, why did that characteristic change? So if we look at the example of the giraffes, they were all, according to Lamarck, they were all born with short necks. And once they had eaten all the leaves and the lower leaves of a tree, the nice juicy new leaves were higher up in the tree. And for them to reach them, they had to stretch their necks. And when they stretched their necks became longer. That then became the acquired characteristic. And it became longer through use. Okay, so they used their necks to stretch. And that then led to it becoming an acquired characteristic. So the challenge then was to reach the food higher up on the tree. Okay, then what did the organism do? So they stretched their necks, and what characteristic was then acquired? And that was the characteristic of long necks. What was the result? Well, that characteristic of long necks was passed on to the offspring, and that is why giraffes all have long necks. Okay, so those are the kind of things when we have a look. And I know the giraffes is a simple example. And there are very many examples, and sometimes the examiners give really difficult examples. So this is why it is important that you use past papers to revise these questions. Right, now we know that it's not the phenotype that affects the genotype, or it's the phenotype that is inherited. It's actually the genes or the genotype. So there's no evidence that acquired characteristics are inherited by offspring. Okay, and that we must just remember. It's the same as if somebody had to lose a leg in an accident, and then they have children after that. There won't be children born without a leg. Okay, that's not possible. So, organisms then did not evolve because they wanted to. According to Lamarck, he said that there's no organisms nobody wants to die, nothing wants to die, they want to live forever. So he believed that organisms chose, so those giraffes chose not to starve and die by stretching their necks to get to the leaves on higher up in the tree. We know that change comes through random mutations, and that is how an organism or populations would change, their genes would change. All right, so we know an organism cannot change its DNA. So if I'm tall and I want to be short, I can't change my DNA. That's who I am. And according to Mendel, we now know that it's the genotype that determines the phenotype and not the other way around. And this is why Lamarck's theory has been rejected. All right, now the second theorist that we learn about is Charles Darwin. And he also worked in the 1800s, but he worked a little bit later than Lamarck. And what he did is he based his theory on observations when he traveled around the world. And he then postulated the theory of natural selection. And his theory basically has two main points. First of all, that species were not created in their present form. Okay, and we said earlier, they evolved from ancestral species. And if we have a look at this diagram, and it's hard to believe, but today what we know as a whale and even a dolphin actually originated from a four-legged land mammal or animal that looked like a dog. Right, but they then, it's that organism that evolved and is known as, has come to be what we know as the modern whale. All right, so species were not created in their present form. And secondly, he proposed that there was a mechanism that caused this change, and that he named natural selection. Right, natural selection, there's 3 points or 4 points, and firstly that there must be variation within the population. So we have different forms, then lots of offspring are produced, there's competition between those offspring. Those with the favorable characteristic, they will then be selected or they will survive, and that is why we see as time goes on, the population changes, and we see more of the organisms with the favorable characteristic in the population than those that don't have the favorable characteristic. Right, if we have a look at punctuated equilibrium quickly, and basically that theory was postulated by Aldridge and Gould in 1972, and basically they explain the speed at which evolution takes place. So they said that there were long periods of time, and if we can see here where there's little or no change, and then suddenly in a very short space of time there's quite a lot of change. Okay, so that is why it's called punctuated equilibrium. All right, so here we see the different example or the different versions of explanation of evolution, and the one is gradualism. So here you can see from the original organism, the changes gradually occurred until we see we have what we see today. Okay, so we see there's two different compared to the ancestor species. However, in punctuated equilibrium, there's rapid change. So here we see the ancestor. Then there's a very short period where we have rapid change. And then there's a long period of no change, and then we see a short period of rapid change again. Yes. All right. Then we're going to have a look at artificial selection. And please, this cannot be, or it shouldn't be confused with GMOs or any kind of genetic modification or biotechnology. Now, in artificial selection, it's the intentional reproduction of individuals in a population that have a specific characteristic, and usually it is to fulfill human needs. So here we can have a look here at the different chickens. You can see how their weight has increased over time, and obviously we're going to reproduce with organisms that are bigger, because then that means more food and more money. Right, speciation. It's very important to know the terms, and here we must know the difference between population and species, and we can't use those terms interchangeably. A population is a group of organisms in one place at a particular time, whereas species is the whole group of that type of organism. Okay, and they obviously can't be all in the same place at one time. Speciation is the process where we actually create new species when a population becomes divided. And the division can either be a geographic barrier or it can be a reproductive mechanism. All right. So speciation is the evolutionary process by which populations evolve to become distinct species. Now, usually we explain speciation through geographic isolation. So we need to say, and this is why I say we need to know the difference between population and species. So we use the term population, and it should be split in two. There is no gene flow, so they've been separated. Wait, let me first say, the population is split into two, either by a river, a sea, a mountain, a lake. The examiner in the question stem will give you whatever has divided the population. And then because they are divided, there's no gene flow. They're not able to interbreed. So they become now reproductively isolated. They then are exposed to different environmental conditions. And it's the environmental conditions that will select certain characteristics. And remember, they're not cross-reproducing. So mutations are going to happen randomly in each population too. So they both undergo natural selection independently. The process is not the same in the two populations. So they become genotypically and phenotypically different. And even if we brought them back together, they would not be able to interbreed. We now have two different species. Right, then the examples that you need to know that are listed. We have in animals, we have Galapagos finches, and we have the Galapagos tortoises. So those are examples in animals. If we have a look at, again, I said to you about the flightless birds. We also have other animals that we find on the different continents that are similar. We have plants. If we have a look in South Africa, we have the baobab tree. But if we have a look in Madagascar and Australia, there are also different species of baobab, which means that the continents must have been together. All right, and then the second example is of our national flower, which is the protea. Australia also has proteas. All right, then the mechanisms of reproductive isolation. Here you just need to know, and this comes straight out of the exam guideline, the different forms of, or the different mechanisms. So the first one is breeding at different times of the year, species-specific courtship behavior, plant adaptations to different pollinators, infertile offspring, and prevention of fertilization. Okay, and there are the examples. Right, evolution in present times. So we use organisms that are rapidly reproducing, and we can see evolution occurring. And one of them is currently affecting us at the moment, which is the SARS coronavirus, which we know about the mutation. So we see different strains. All right, we're going to have a look then quickly to finish off at human evolution and what you need to know. So these 4 points are very important. You need to know phylogenetic trees. You need to know the similarities and the differences between humans and African apes. You must know the lines of evidence. So here again, we're looking at evidence for human evolution, and then we're going to have a look at the out of Africa hypothesis. Right, now to interpret a phylogenetic tree, this is an example here, and you can see this is the hominin family that is simplified or is being placed on the tree. It's a very simplified version, and that is the classification of humans, for example. Okay, so the possible questions that you can get is an interpretation of a phylogenetic tree. They can give you similarities between African apes and humans. They can ask you for differences, where we look at the different skulls, or we can look at the difference in the pelvis, or different parts of the skeleton. Then we also look at fossil, genetic, and cultural evidence, and then the fossils that were found in South Africa. Those ones you must know. And then the out of Africa hypothesis. All right, a phylogenetic tree. And remember earlier I said to you, you need to know the difference between a pedigree diagram and a phylogenetic tree. Please do not mix these two up. A phylogenetic tree is a schematic form that shows the evolutionary relationships between organisms. So we can see how closely they are related, or how far or how distantly they are related. Now the term phyllo means the organism's phylum group, and then genetic is obviously from the genes. And remember I said to you, when we look at genetic evidence, we'll have a look at how much similarity there is between the genes or how much difference there is. And that's how we then can determine how closely they are related. Right. Okay, so, and then also very often in a phylogenetic tree, when you are being asked on it, they very often ask who the common ancestor is. So remember, just before the branches split, the organism that was just before the split would be a common ancestor of the two organisms that we find on each of the branches. All right. So if we have a look at the phylogenetic tree on the right, the first branching or speciation event is at number 1. So there we can see at number 1, there was a speciation event, and it split into 2 branches. Then we have a look, and so number 1, that is where the common ancestor would be, actually of all the species that we have at the top. Then we have a look further up. We can see over here at number 2, this is the common ancestor of these 3 organisms. Okay, and here again there was another speciation event, so number 3 then would be the common ancestor of B and C. So this is the way in which you would interpret a phylogenetic tree. And if we asked, we can see here that these organisms on this side are not as closely related to the organisms on this side because the speciation occurred right back here. Okay. All right, so always remember, just before the branch splits into two, that is where we find the common ancestor. And then the two branches, the two lines, or the two, the two lines that the inherit or the evolution follows, those then are known as the descendants. Right, so we can have a look here and we can see further on again, here we had another speciation event where another species branched off from the original. And then what is important also is to notice that wherever the line stops, if it doesn't stop at 0, that means this species has then become extinct. Okay, so that indicates extinction. However, here we see this species is right at 0, which means that they still exist today, and that means that they are extant. Then, where, as I've said earlier, where a branch splits, that is usually known as a speciation event. And remember, we looked at speciation and we said for some reason the population is split, and then over time they become different species. Right, then the similarities, and this is very important, you must know them. The first one is where between humans and African apes is that we have large brains or skulls compared to the body mass. Also the eyes are in front, we have binocular vision or stereoscopic vision, freely rotating arms, long upper arms. Okay, it's not the upper limb, because remember your whole arm is your upper limb. It's just the upper part that is lengthened. All right. Then we have elbows that rotate freely. Okay, and they can rotate of the forearm. We have 5 fingers, and there on our hand, one of the 5 fingers would be your opposable thumb, and as you can see, it's opposite, and it allows us to grip or to handle things. And then another difference, also still on the hand, is the fact that we have flat nails, we don't have claws. Okay, so that's a similarity. The next similarity is that there's 2 mammary glands, and then the eyes have cones, or we are able to have color vision, so we are able to see the difference between colors, whereas other animals would only have monochromatic vision. All right, and then there's also sexual dimorphism. So between African apes and hominids, you can see that there's a difference between the male and female. You can see. Okay, so you can see the difference. All right, so it's very important that you do know these similarities. They are frequently asked. They might not ask you all of them, but they could ask you the similarities of the skull, for example. And then also remember in your terminology, you need to know the difference between what is the skull. Okay, and remember the whole structure that is the head is the skull. That includes the facial bones. When we talk about the cranium, we're only talking about this part of the skull, and the cranium then is basically the bone or the part or the structure that houses the brain. Okay, so there is a difference. All right. Now when we look at the differences between humans and African apes, we know that humans are bipedal. And remember, bi means 2, and whereas African apes are quadrupedal. Quad means 4. So humans walk on 2 legs all the time, whereas quadrupedal organisms would walk on all 4 limbs. Now there is a difference in the structure of the skeleton, because of the different modes of movement. So the first one for humans that are bipedal, the foramen magnum, which is a hole in the skull that we find that is allows the spinal cord to leave or actually the spinal cord to enter and join with the brain. And that position then is usually in a forward position. We don't talk about it being in the middle or in the center, because this could be the middle, this could be the middle, that could be the middle, anywhere. So we normally talk about in a forward position. Whereas in quadrupeds, the foramen magnum is usually towards the back of the skull. Then if we have a look at the spine, we find that humans have an S shape. Their spine is curved, and we talk about this as being more curved than that of a quadruped, okay, because they're going to walk on all fours. Right, then also another difference is a shorter and wider pelvis. So humans have a much broader and narrower, so it's shorter pelvis. It's quite bowl-shaped, and that is to carry the weight of the upper body, whereas the African apes have a longer, narrower one. The cranium size, as we said, is larger, whereas this one is smaller. Palate shape, this is semicircular, whereas this one is more U-shaped. Dentition, we find small gaps or no gaps between the teeth and much smaller canines, whereas here we have large gaps and very large canines. A flat face, they are non-prognathous, whereas in African apes the face is prognathous. And the cranial and brow ridges in humans is absent, there aren't any, whereas they are quite distinct in African apes. Alright, so those differences you must study. Right, then the lines of evidence is fossil evidence, genetic evidence, and cultural evidence. Okay, so we have a look to see the similarities in the fossil structures. Genetic evidence, again, we also look at the similarities or the differences, and then we can have a look at the cultural evidence. So the use of tools tells us how far or how more evolved organisms or humans are. Okay, then the fossil evidence in South Africa that you must know these ones. The first species is Australopithecus africanus. You must know the tongue child, where it was found and who discovered it. Mrs. Pless, where it was found and who discovered it. Littlefoot, the same thing. And then the next species is Australopithecus sediba, also commonly known as Karabu, and where that was discovered and who. Then we have a look at Homo naledi, and when and who, where they were and who discovered them. And then the last example is Homo sapiens, which is humans, which is us. Okay, then the evidence for out of Africa hypothesis. Again, we said there's fossil evidence. So we have a look, and this is straight out of the exam guidelines. And what you need to do is when you answer this type of question, you answer it specifically. So you need to say, Ardipithecus fossils are only found in Africa. Australopithecus fossils are only found in Africa. So that is very important, that they are only found there. Then all the Homo fossils. So we find Homo habilis, we find Homo erectus. We do find them elsewhere in the world, but the oldest ones are found in Africa. And all of this is evidence that humans originated in Africa and then migrated to other parts of the world. We also look at genetic evidence, so we look at the mitochondrial DNA, and remember that is inherited down the maternal line, and we analyze the mutations on the mitochondrial DNA, and that's how we can trace where the oldest ancestor comes from. So the individual with most or more mutations will then be older than the individual with fewer mutations. And wherever they are found, that means that the oldest individual originated in a certain place. Right, things to take to the exam room. Very important. The first thing is a ruler, because we've used it in several ways. The second thing is a compass to draw circles, and that is if you are asked to draw a pie chart. And very important there too is the protractor, so that we can work out the degrees of the segments that you need to draw. And then the next thing is an eraser, so that you can erase if you've made a mistake. A calculator to do calculations, and then obviously a backup pen. You take your pen and a backup, and you can write either in blue or black pen, and so that you are well prepared for the exam. So I just want to say thank you very much and good luck. Use all the resources that you have. Ask your teachers for, if you don't have past papers, there are exemplar papers as we spoke about. There also is a website where you can get a lot of the documents from. So good luck for Monday, and I hope it all goes well. Thank you for watching the lesson. Please remember, sanitize, wear your mask, maintain social distancing, and keep safe. And we hope you do well. Thank you. Goodbye.